Our next speaker is Silvere van de Maarel to speak about the genetics and genetic testing for FSHD.
First of all, I would like to express it's an honor and a pleasure to be here again at this patient conference. It's always a joy to be here and to interact with patients. I'm a geneticist, preclinical scientist, so I don't interact that often with patients, and for us it's always very rewarding and enthusiasmating to talk to you. So I'm based in Leiden. Here you see actually the research building that we are working in, and our hospital has a very long tradition in the study of FSHD. Let me see whether I can get this to work. Yes. Which started in the 80s with Professor Patberg and France, who decided to work on this disease, FSHD, that seemed to run in families, but we had no clue how that was actually working. So from that moment on, we started to collect DNA and other materials from patients and starting to identify the genetic defect in FSHD. And we're still doing a lot of genetic studies in FSHD, and here you just see some photographs of storage of DNA samples and the processing towards the genetic confirmation of FSHD. Nowadays, we also do many studies on the molecular and the cellular biology of FSHD, and our hospital is the only center in the Netherlands that performs the DNA diagnosis for FSHD. We also assist in the diagnosis of FSHD since we feel, so we do that for about 250 to 500 cases annually from all over the world, since we believe the only thing that we can offer patients is a good genetic diagnosis. So if we can assist in that, we will do so. So I've been involved in the science of FSHD since 1997, and really for many, many years, we only had one question, and that is, what is causing this disease? Already in 1991, our group identified the main genetic defect, this reduction of the number of repeat units on chromosome 4. But we really had no clue how that actually could cause a muscular dystrophy. And I think it's around 2010 where we as a research community really identified a plausible disease mechanism. And from then onwards, we really are much more focusing on how can we actually stop this process. So some of the slides that I will be showing you are somewhat redundant with what you just heard by Alexandra Balayu, but it's a complex situation, it's a complex matter, so perhaps it's not so bad it's even complex for us, so it's sometimes good to hear something from a different angle and different perspective. What I show you here is just a simple, basic biology that we all need to learn a little bit, know a little bit about to understand the process. So you heard Alexandra talking about DNA, which is basically providing the cell with all the information it needs. It's the library that encodes the genes, the recipes for all the cookies. Now, this DNA you don't want to mess with. You know, the first and most prominent function of the cell or task of the cell is to keep the integrity of your DNA, because this is basically where all your information is stored. So it never leaves this small nucleus there. There are two important aspects to DNA. First, you need to be able to copy it so that the daughter cell has exactly the same information as the original cell, and you need to use information from it, so to make your proteins, to make your cookies. And as was alluded to you in the previous talk, the way we do that is by copying pieces of the DNA, the genes, and creating the proteins from that. Now, this is a formidable task to the cell. And also, this was already shown a little bit, but you really have to realize that within each nucleus, there is physically over 6 feet of DNA. The nucleus itself is only 10 micron. So it's really the same as putting half a marathon in a tennis ball. This is what every cell does in your body. It's a formidable task. So the way it can do so only is by wrapping it up very, very tightly, as I've shown here in the bottom of these springs, where you really wrap it up very tightly. The consequence of that is that you can't access the information anymore, right? Where it's so tightly packed, you can't read it, you can't copy it. So the cell needs to make clever decisions here. Unfortunately, the cell can do so by actually changing the structure whenever it needs to do so. So it basically can put your recipes in a more open structure of the spring or in a more closed structure of the spring. So in the on or off situation, as I'm showing here in the spring situation again. And it makes sense, because we only have one genome, right? Every cell in our body has exactly the same information. Yet we all know that a cell in your liver is functionally and morphologically completely different from a neuron. So there are about 20 to 30,000 recipes in our genome, and the cell doesn't need to use all of them. It can selectively use only those that it needs to be either a cell in your liver or a neuron or a skin cell. So this is how it actually works with the DNA, with the information. It only puts that part of the information that it really needs in the open part of the spring, while all the other information that it doesn't require, it puts away in the closed part of the spring. And this is where it goes wrong in FSHD. Because in muscle cells, this repeat structure on chromosome 4 belongs in the closed part of the spring. The information that is in there should not be used by the muscle. It doesn't need it. Actually, if you use it, it's actually damaging to the muscle. So normally in individuals with long repeats, this part of the repeat is in this closed environment of the spring. However, if the repeat becomes too short, and this is in the majority of individuals with FSHD, the cell can't manage this anymore, and the repeat ends up in the more open structure of the spring. Hence, the information becomes available, and this DUX4 protein is being produced. This crazy chef from Alexandria is being produced and causing damage to the muscle. Now, there is a second form of FSHD, which is much more rare than FSHD1. So FSHD1 accounts for more than 95% of all the patients. And in FSHD2, it's not the repeat itself, but it is a protein that you need to maintain this repeat in the closed environment of the spring. If you lose that protein, again, your muscle is not able to maintain this position, and again, the repeat ends up in the more open situation, in a more open place of the spring. Again, same consequences. Suddenly, the information that is embedded within the repeat becomes available to the muscle. It starts to produce DUX4. Recently, we have shown that although since we were not able to explain the genetic mechanism in all of our patients, that another protein, which you also need actually not to maintain it in the closed part, but actually to put it in the closed part of the spring, that if you have mutations in there and therefore reduced activity of that protein, which is called DNMT3B, you actually end up again in exactly the same situation. Again, the repeat moves to the open part of the spring and DUX4 can be produced. Now, this is a very rare condition. Thus far, we have only identified 2 families with mutations in this gene, but I'm expecting that now this information is known that we will see more of these families. Now, this is a plausible mechanism. All the patients converge at this one single phenomenon that the information that is in the repeat becomes available to the muscle, and this protein DUX4 is produced. This doesn't happen on all chromosome 4s, and it was also shown by Alexandra. It all depends on a very small variation that is immediately distal to the repeat on the left side for you of the repeat, which is called a polyadenylation signal, or a PAS. This is essential for the DUX4 protein to be produced. However, it's not present on all chromosome 4s. It's only present on half of the chromosomes 4. So only if you have the loss of this control over the expression of DUX4 in combination with the presence of this PAS, of this polyadenylases signal, it will result in the production of DUX4 protein in the muscle. If it's not there, then you will have the opening of the structure, you will have an attempt to produce the protein, but it won't be a stable situation, so there will be no DUX4 produced. So, as I said, we have developed a plausible disease model for FSHD. There's a gene that's called DUX4, and it's encoding a protein which is normally not expressed in muscle. If you have a contraction, so reduction of the D4C4 repeat numbers, or if you have mutations in proteins that you need to keep this environment closed, then you get the production of DUX4 in the muscle, and that leads to toxicity to the muscle. And although that's a very appealing model, it doesn't explain the variability in onset and progression of the disease. And this is something that you are much more aware of than I am, but you all experience within your families that some individuals are much more affected, show a much more progressive, much more progression than other individuals that actually have exactly the same mutation. And it's already known from the very first description of this disease by Landouzy and Jugerine, two French neurologists, in 1894. And here you see two nephews. I believe these are two nephews. You see they're about the same age, and the upper individual is much more severely affected than the bottom individual. So we started to think about this concept. Can we actually explain some of this variability at the genetic level? And the bottom line is yes, we can. And the reason for that is that we're starting to understand that you have an interplay, a combination of the size of the repeat and the proteins that you need to repress the repeat. So basically what we have found are families where you not only have a reduction in the repeat number, so a typical FSHD1 situation, but these individuals also have mutations in SMCHD1 or in DNMT3B. Now, these two mutations together act synergistically, and therefore the spring becomes even much more open, the information becomes much more available, and much more DUX4 is being produced, leading to a much more severe phenotype. Now, I think this is an important observation, as we all differ a little bit in our ability to repress DUX4, and in our ability in the activity of SMCHD1 and DNMT3B. So we believe that this actually may explain some of the variability that we see within families. So in summary, we have a very good view now on the genetic causes of FSHD, and it is very well diagnosable. It's either a contraction, so a shortening of the repeat on a 4qa chromosome that is in more than 95% of the patients, or mutations in SMCHD1, and in very rare occasions, mutations in DNMT3B. That needs to be combined with a 4qa chromosome. We also need to realize that there are still patients out there that we cannot explain, so there are likely some more mechanisms out there. And there are also individuals who have a phenotype that looks a lot like FSHD, but we call FSHD syndromes or mimics because they have mutations in different pathways and different proteins that are not related to DUX4 itself. Now, an important other observation is the question, can we actually reverse this situation? So I told you that the spring is opening up. Can we close it again? Well, in muscle cell cultures, yes, we can. We can actually add SMCHD1, and if you do so, you can actually repress DUX4 again. So there is a reversibility to the situation, which is hopeful in the terms if you think about therapies. So there are many therapeutic strategies out there now that are being pursued, and they act on all levels that we know about the disease process now. So can we either prevent or restore the closed structure of the repeat? Can we prevent the production of the RNA, the DUX4 RNA, or the DUX4 protein, or can we perhaps prevent the action of the DUX4 protein itself? And finally, we can also think about, since this DUX4 is this master chef, can we block the individual pathways that the DUX4 protein is acting on? And with that, I would like to thank all the people in my lab who are doing the work and my collaborators and the funding organizations that are supporting me. Thank you.